I'm going to mute, so everybody should mute except for Donna, and then I'll hit broadcast.
All right.
Good luck, everyone.
Good luck.
Last one.
I know.
Kind of sad. All right, I'm going to broadcast, Donna.
Okay.
Good luck.
Thank you for joining us for the last Friday evening lecture of the 2020 virtual lecture series. We'll get started in a couple of minutes. We're just waiting for a few more folks to join us. Dr. Nipam Patel will give a brief introduction to the lecture, followed by an introduction by Dr. Stephanie White, and then Dr. Jarvis's lecture as well. The lecture will be followed by a live Q&A, and you can submit questions at the bottom of your screen in the Q&A tab during the lecture, and we will try to get to as many of you as we can. And again, we'll just wait another minute or so. I can see people coming in. When you submit a question, please let us know where you are watching the lecture from so that we can know how far we're broadcasting tonight. I'll just wait another second or so. And remember, you can submit questions in the Q&A tab at the bottom of the screen during the lecture as well as after the lecture, and we will try to get to as many of them as we can.
Good evening and welcome to tonight's Friday evening lecture. I'm Nipam Patel, Director of the Marine Biological Laboratory. The MBL is dedicated to fundamental biological discoveries that inform human health and the health of our planet. Every summer since its founding in 1888, the MBL has hosted public evening lectures where renowned scientists, historians, and architects of modern biology have presented stunning scientific discoveries for exploration and discussion. Despite the hardships of the war years and the Great Depression, the Friday evening lectures have always been held without interruption. Just as our predecessors did, we are coming together as a community to adapt this year's series with streamed lectures delivered by notable Friday evening lecture alumni. While I regret that we can't be together in person in the Lilly Auditorium, your presence here tonight is helping us to keep this important tradition alive. Thank you for joining us and enjoy the presentation. And remember to turn off your cell phones.
Welcome. I'm Stephanie White, Professor of Integrative Biology and Physiology at UCLA and co-director, along with Alberto Pereda, of the Neural Systems and Behavior course. Alberto and I also have co-directed the GRASS Labs at the MBL. Although many MBL in-person courses and events had to be canceled this summer, like many of you, I've been avidly following the Friday evening lectures. Despite the pandemic, the series remains outstanding, and the numbers of virtual attendees has far surpassed those who could comfortably cram into Lilly Auditorium. Tonight, I'm delighted to introduce the final speaker in this series, Professor Erich Jarvis, coming to us from the Laboratory of the Neurogenetics of Language at the Rockefeller University, where he's also director of the Field Research Station in Millbrook and the Vertebrate Genome Lab. I'll now give a brief synopsis of Dr. Jarvis's academic story. He grew up in New York City and attended the High School for the Performing Arts, as profiled in the popular movie and TV show Fame, where they say, dream big, think big, reach for the stars, be ambitious, be bold, and don't let anybody stand in your way. Dr. Jarvis is the embodiment of this advice. He received scholarships to the Joffrey Ballet and Alvin Ailey Schools of Dance, but chose to go into science, double majoring in math and biology at Hunter College under the mentorship of molecular microbiologist Rivka Rudner. As an undergrad, he published 8 papers, including 2 as first author. To all undergrads out there listening in, Dr. Jarvis attributes these accomplishments to motivation or to getting a high from an experimental result, much as he would from nailing a dance move. He also emphasizes discipline, which of course means practice, practice, practice. These themes may have influenced his choice to join Fernando Nottebohm's Songbird Lab at Rockefeller for his graduate work, where he stayed on for his postdoc. There he made a major discovery of motor-driven gene expression. In brief, Dr. Jarvis showed that gene activation in the songbird brain depends on whether or not there is an audience listening in to the song. Later, as an assistant professor, he showed that singing activated an entire cascade of gene expression in song-dedicated brain regions. Dr. Jarvis was recruited to Duke University as an assistant professor, where I first met him. There he secured funding to convene a group of comparative brain researchers worldwide to correct wrong assumptions about avian intelligence. Attending it was like being at the United Nations of renaming the bird brain. The resultant paper reconciled outdated names and overlooked or newly recognized cognitive capacity of birds and has 1,000 citations. These early accomplishments are a prelude to his becoming the co-leader of an avian genomics consortium consisting of over 200 scientists in over 20 countries to oversee sequencing of genomes representative of every avian order. As another example of his leadership strength, Dr. Jarvis is the only scientist ever to author or co-author 8 publications in a single edition of Science magazine. One of these papers highlighted the extreme molecular convergence between humans and songbirds within specialized brain structures that control speech and song, respectively. Dr. Jarvis has received many awards throughout his career. I will highlight only a subset. He was an NIH Mark Scholar as an undergrad and a Ford Foundation Fellow as a grad student. As an assistant professor, he received Klingenstein, Whitehall, Packard, and Frontiers in Science Awards. Perhaps most notably, he was a National Science Foundation Alan T. Waterman awardee, an honor given to just a single scientist in any discipline in any one year. He was an NIH Directors Pioneer, an Ernest Everett Just awardee, and is now a Howard Hughes Medical Investigator. Erich visited the Neural Systems and Behavior course several years ago, and he fit right in due to many of the attributes I've already described. Like Erich's alma mater, courses at the MBL encourage students to dream big, think big, reach for the stars, be ambitious, be bold, and don't let anybody or anything like time or technology discourage you. With that, I invite you to enjoy Dr. Jarvis's lecture, which is entitled Molecular Convergence in Brain Regions for Song Learning in Birds and Spoken Language in Humans.
Well, it's a pleasure to be here, or to be here present online to give an MBL Friday lecture. I gave one back in 2008, and it was a great experience back then, and I hope it will be one too. And what I'm going to tell you about is where we've come from since I've last been giving this lecture. The title of my lecture is Molecular Convergence in Brain Regions for Song Learning in Birds and Spoken Language in Humans. And so what does all that mean? Well, I'm interested in understanding how the brain produces complex behaviors. And one of the behaviors that is the most complex ones for us is spoken language. And I've been fascinated by why we humans can do this, and only a few other animals, but nobody else can actually produce what we call learned imitated sounds. What I'm going to talk about today is things that we have learned over the last 10 years or so about the actual genetic molecules, the genes, that is, that actually construct the brain circuits for speech and also vocal learning in other species. So to get into this, in my first slide here, I just basically showed the outline. We have, we're going to talk about this from the level of the actual behavior itself, the anatomy, and the genetics, and then a hypothesis that we're going to put forth in explaining how did spoken language evolve in humans and songbirds. And I encourage you to get more background on this subject. At the end of last year, published an article titled Evolution of Vocal Learning and Spoken Language in a special issue in Science magazine on this subject. That is the science behind brain and language. So I like to think of language as has multiple components to it. This is called the multi-component view of spoken language. It's not me who actually came up with this idea, but in this slide here, it's my view of how actually you break this down. Many of us, when we experience language, and spoken language in particular, we don't realize that there are multiple components to this ability of what we do, that we share some of those components with other animals and others not. And here I have listed 7 such components. One is called auditory learning, which is very common in many different species, but another highlighted in red here is vocal production learning. And that's the ability to produce novel sounds. And all of these things like syntax, vocal usage learning, and so forth, make up what we call spoken language. So what about this one rare ability that I told you about, the vocal production learning? Well, that's only been found in 5 groups of mammals, like humans, dolphins, and whales, who are together called cetaceans, bats, elephants, and seals amongst mammals, and parrots, songbirds, and hummingbirds amongst birds. Now, all 8 of these groups of species, of the mammals and the birds, all of them have close relatives that don't learn how to imitate sounds, like chimpanzees to humans or falcons to parrots. And so it's thought that all of us evolved this ability independent of our common ancestor. But vocal production learning is different from the other kinds of components I mentioned that is more ubiquitous. Auditory comprehension learning is more ubiquitous. And here I give an example where you can tell a dog, or you can teach a dog to learn how to respond to the human word sit. And the dog will sit, or siente say in Spanish, osuade in Japanese. In any language, you can teach a dog the meanings of human speech sounds. But a dog can't say, okay, you got it, I will sit, or a dog tell you to sit. Instead, a dog says woof, or barks, and so forth, for what it wants. And those sounds that a dog makes are innate vocalizations that he can't learn to modify that much, but he can understand our learned vocalizations, and we call that auditory or comprehension learning. A dog can learn how to use his innate vocalizations for specific contexts, like barking for food, barking for attention, barking to go outside, but the actual sounds are all the same. So, but in contrast, the species that have this vocal production learning besides humans, some of them are actually more complex than what we see in many non-human animals. And the example I like to give is parrots. So here in this video that I'm going to play, we have a parakeet called rattle off at least 30 or so words in human speech, whole sentences, recombining sounds into new meanings that he didn't actually learn from his human owners. And when he sees a dog, he actually says meow. So he's in the right category, just says the wrong thing. And he says, I'm a doctor. So Disco, although lots of times is rambling off these sounds randomly, that dog that he says something to can't actually do any of this. And so what about our closest relatives? Every once in a while, there's some great ape or monkey that's doing something that gets people excited. And here is one example. Coco the gorilla.
How about when you're coughing? That was good. You did sneezing and a cough. Excellent.
So this is Coco, who's been raised with humans for 39 years and has good auditory learning, auditory comprehension. That is, you heard Coco respond to the person asking, how about coughing? And Coco actually purposely and voluntarily tries to cough. But that's the best kind of sound you can get out of Coco voluntarily, besides regular gorilla regular gorilla innate sounds. And so there's good auditory learning here, but not vocal production learning. So what this has led to is the questions from us scientists in the slide here that says, we try so hard to get some of our closest relatives, like this monkey here sitting in a chair, to say the word apple, yet this parrot here, 300 million years distant from us, from a common ancestor, can go one better and says golden delicious, or all kinds of different apples, whereas a monkey just goes, huh? What's going on? So what we scientists have been asking is the question of what makes the difference in the brain, the circuitry, or even the periphery about these species that allows us and parrots and songbirds to imitate sounds and communicate in that way, and not our closest relatives. So in my second part of the talk, I'm going to talk about what I think had happened and the motor theory of how this came about. And in that article in Science Magazine, I review 6 ideas or common views out there that what makes us humans and parrots different from these other species that can imitate sounds. And I'm not going to talk about all 6 of them in the talk, but the 3 that I've highlighted here in red is what I'll talk about. But just to quickly review, number 1 is that there's a general difference in the brain, bigger brains of humans, bigger brains of parrots, and by virtue of having more neurons in the brain, we have the more complex behaviors like speech. Or there's a difference in the vocal organ, that in us we have a lowered larynx, and this allows us to make more sounds compared to other species. Or there's a difference in the forebrain, the presence or absence of a brain circuit that controls the vocal organs, is the third one. Or the fourth is the vocal circuit is there in all these species, but in humans and in parrots, it makes a direct connection to the motor neurons that control the voice. And by access of direct connections from the cortex, allows you to have more complex vocal behavior. Or that all species had the vocal motor cortical regions, it's just not connected to the hearing areas. And only in humans or some of these vocal learning species, you get this connection between the two, and therefore that leads to more abilities to imitate the sounds that you hear. And lastly, coming from the linguistics field, hypothesis 6 here, is that we humans have a language module that processes all these complex algorithms for language and then tells the speech circuit how to speak or how to produce those sounds, tells the hearing circuit how to perceive them. And so the ones that have the most support, I believe, in the literature and from our own work is that there is a general difference in the brain. There is a presence or absence of a forebrain circuit as well that controls the behavior, and the direct connection to the neurons that control the voice. And so here, in the next slide showing here in songbirds is supporting the hypothesis of this presence versus absence of a brain circuit. So what we found many years ago is when a songbird sings like a canary, that act of singing shown here is associated with a rapid increase of gene expression in the brain of genes that are responsive to neural activity. And what you see here in this white signal is the upregulation of the messenger RNA of a particular gene we call EGR1 in the brain circuits that control the vocal behavior. The red staining here of the brain is basically a general stain for all neurons in the brain, and the white signal here is the mRNA. And you can see in this graph, the brain sections from this canary show that the more a bird sings, the more increased gene expression you see of this EGR1 gene in the brain, such that for every half hour of certain amount of singing, there's a one-to-one correlation between the amount of singing behavior and the actual gene regulation. And this was a motor-driven gene expression response in the circuit controlling the learned sounds of the canary, such that if the animal's deaf or if it doesn't have any somatosensory feedback from the muscles to the brain and so forth, you still see this singing-driven or motor-driven gene expression in the brain. And we use this molecular mapping tool to ask the question, can we find similar molecular active brain circuits in other vocal learning species and non-vocal learning species? And shown here, we find that all 3 species of vocal learning birds have a forebrain circuit that becomes active when hearing sounds, shown in blue. And you can find that same forebrain circuit for hearing in the non-vocal learning bird species. Here, where it says in blue, where it says non-vocal learners. And actually on the screen behind me as well, you can see that model. Whereas those species that have vocal learning, you find that only they have brain areas in the forebrain, that is active in the production of their sounds, and shown here in yellow and red. And the ones in yellow are the brain pathways responsible for producing the learned sounds, and the ones in red are responsible for actually learning how to produce the sounds. And what's remarkable here, if you look at this phylogenetic tree to the left that we generated with whole genome sequences, it shows that the vocal learning species in the red dots here are distantly related to each other, and they have close relatives to each other that don't have vocal learning behaviors. So what it would suggest is that vocal learners have independently evolved 3 times 7 such brain structures in red and yellow here in the last 65 million years that you cannot find in the non-vocal learners, a remarkably similar brain pathway. So either there are 3 independent gains, or shown by this arrow here, there would have to have been one common ancestor back 65 million years when the dinosaurs were around, and their common ancestor evolved the vocal learning circuit and was lost at least 10 times in a bunch of other bird lineages like falcons and penguins and swifts and nightjars and so forth. So we think it's really 3 independent origins of a similar brain pathway. What about humans? Well, here we show a songbird brain compared to a human brain. And the first lesson you learn here is that brain size does not matter. Here we have a songbird brain to scale with a human brain, and you can see we can fit about 3,000 songbird brains just into one human brain. Yet, a brain bigger than these songbirds, like a chimpanzee, can't do what these songbirds do. The second lesson we learned here is that cortical folding doesn't matter. Here in the human brain, we have lots of cortex folding, whereas the songbird brain, the cortex region is smooth. So I think the real difference is the presence or absence of a circuit. But to get at what that circuit looks like, we had to challenge a century-old view of understanding brain organization. So here shown avian mammalian brain homologies. It used to be thought that the human brain had these ancestral reptilian-like basal ganglia structures near the bottom of the brain, and they're called basal ganglia. And it was thought that these brain structures evolved in a linear-like fashion with the purpose in mind, the eventual purpose to generate humans. That is, the early comparative neurobiologists, the patriarchs of neuroanatomy over 100 years ago, viewed evolution as its purpose was to generate humans, not as Darwin viewed it, in a tree-like fashion, but in a linear fashion, and particularly to generate Europeans, with Europeans having the biggest brains. So you can imagine the kind of thinking they had at the time. And they viewed that the fish and amphibians and reptiles and birds all were adding new structures on top of each other, in purple here, to eventually generate the mammalian brain with this new neocortex in green here. We now know that this whole idea was false, and the real truth is that bird brains have a large cortical territory just like humans, just like other mammals. In green here, it's just not layered. It's nuclear in its organization. And the different nuclear cell types correspond to different cell types in the layers of the cortex. And so putting all that together here, what we find is that when we take that same model, like that behind me, of these vocal learning birds, and now compare it to what we know of human speech areas, and what we know in non-human primates, we can see that all mammal species, all bird species, and I'm going to argue all vertebrate species, have what I color-coded in blue here, an auditory pathway that's responsible for that auditory comprehension learning, like allowing dogs to learn human speech words and respond to the sound sit, or Coco the gorilla to hear someone say, how about coughing, and try to cough. But only humans have this other circuit I've shown in yellow and red here that's responsible for spoken language that is similar and convergent to these brain pathways in the vocal learning birds. And shown in this red arrow, those brain circuits in humans make a direct projection to the structures or the motor neurons in the brainstem that control the vocal organs. You can't find such a connection like that in the non-human primates. So how could this have come about? How could this similarity between humans and the vocal learning birds come about between us, but not in our closest relatives? I didn't have a theory at the time, or at least a hypothesis, until we discovered, as seen in this next slide here, that the vocal learning brain regions seem to be embedded in brain regions that control learning how to move. And how was that discovered? We found that not only that singing behavior causes the activity-dependent genes to light up in the brain, like you see here in white, in this white signal, but the brain regions surrounding these vocal learning circuits light up in the brain when we're learning how to walk, when we're, in this case, the bird hopping in a wheel, and so forth. And we see these motor circuits that have similar connectivity as the vocal learning circuits in all the vocal learning species surrounding the vocal learning areas, but we also see them in non-vocal learning species, but without the presence of the vocal learning circuits inside of them. And so what does that mean? That led to us asking the same question in mammals, and a colleague of mine, Steve Brown, found that when you have dancers, image their brains, like with imaging equipment here, and look at their brain's activity while they're doing learned choreographed movements, you will find that the brain areas in our own brain light up when we are learning how to walk, learning how to do choreographed dance, are directly adjacent to the brain areas that control speech production. So this led to what I call the motor theory of vocal learning origin, where I argued that all vertebrates have this innate brain circuit that controls from the brainstem that controls the dog's barking, the ape's innate calls, or chickens crowing. And that innate brainstem circuit can be found in all species, in yellow and red here. All species, I also argue, have a forebrain circuit here in the green and the purple and the blue areas. One part of that circuit is controlling the ability to make movements. The other part of that circuit is controlling learning how to make the movements, like teaching a dog to learn how to do tricks. And I argue that that motor learning circuit is being replicated multiple times during embryonic development, and it gets replicated one more time in the vocal learning species through some genetic mutation, and that extra replication of the motor learning pathway then synapses on and takes over the brain stem circuit for the vocalizations, and now you get an emerging vocal learning circuit that is similar to the surrounding brain pathways that control movements, learning how to move. And basically speech is learning how to move the muscles that make sound. And so how we are going to test this theory? Can we test it through, from a molecular level? And that's the next part of my talk here. If this hypothesis is true, it means that the brain regions that control vocal learning should have genes that function similarly to the circuit around it that controls learning how to move, and different from circuits that control vision or control hearing in that sense. So how are we going to address that question? What we did is that we laser dissected out the song learning nuclei of each of the vocal learning bird groups and the surrounding brain circuits. that control learning how to move. And the Allen Institute for Brain Science also did something similar for for humans and non-human primates. So from 6 postmortem samples of people who volunteered to give their organs to science after they died, they profiled the genes in 3,700 samples from 6 humans and 252 samples from 6 monkeys. And folks in my lab here, like Andrea Spenning, Irina Hata, Osceola Whitney, analyzed this data of gene expression levels of thousands of genes in the songbird brain and the human brain and non-human primates and non-vocal learning species, and asked, can we find genes that are specialized in their regulation, increased or decreased relative to the surrounding brain circuits? Can we find those genes that might be specialized in these vocal learning circuits that differ from the surrounding ones and those that are the same? And the answer is yes. And what was remarkable is that we found that the cortical areas that we now designated the cortex of birds did match in its molecular profile the cortex of the human brain, getting rid of that 100-year-old century vision that the bird brain is very primitive and doesn't have any cortex. And also, by the way, European brains aren't bigger. That was also gotten rid of. Everybody's brain is really big or smaller relative to their body size, not relative to their intelligence. And even then, there's no difference in intelligence between different ethnic groups. And there isn't much difference in vocal learning intelligence between different species of birds. So what was also interesting is that the brain regions that control learning how to imitate sounds showed molecular convergence with the speech areas of the human brain and the songbird brain. Shown here for the HVC and RA song nuclei match different layers of the cortex of the laryngeal motor cortex of humans. Layers 2 and 3 for HVC, layers 5 and 6 for RA. And an area in the basal ganglia called area X, here in purple, that controls learning how to imitate sounds, matched the part of the human striatum called the anterior striatum that was active during speech production, particularly learning how to produce speech in humans. And this was supported by what we call RNA-seq data, recently, again, repeated work from Gregory Gedman in my lab, who found that HVC matches the layers 2 and 3 of the laryngeal motor cortex of humans. And we cannot find such convergent gene expression changes in non-vocal learning birds like quails or non-human primates like marmosets or macaques. And so what's going on here? Well, it's not just 1 or 2 genes. Like in the laryngeal motor cortex region, we find that 55 genes differ in their regulation in the speech areas, in the laryngeal motor cortex areas for humans and the RA cell nucleus equivalent for the vocal learning birds. And many of them are downregulated, as it's shown in blue here, where all the vocal learning birds have downregulation of these axon guidance genes like SLIT1, plexin C1, GAP43. And humans also have downregulation in the laryngeal motor cortex of these genes. And the ones that are upregulated in this region, like parvalbumin, are involved in neuroprotection, like buffering calcium over toxicity. So what is going on here? Well, a lot of these genes that are downregulated are involved in neuroconnectivity and are repulsive to forming connections when they are at high levels. So we think that preventing connections from forming or allowing certain other connections to form. But why upregulate these neuroprotection genes? Well, it turns out that the speech areas in us and the song areas in songbirds have high firing rates, even when we're not talking. But certainly when we're talking, they have higher firing rates from the surrounding motor areas. And those higher firing rates, I think, can cause neurotoxicity. So we need to upregulate these genes to protect these cells from dying as we speak, so that we can speak another half hour, an hour, or a day later. And so how do we go about testing this? Well, we decided to look at some of these genes that are downregulated, like SLIT1, to try to validate is that true. And here in this slide, you can see the white signal again being the mRNA, that SLIT1, this axon guidance ligand, is downregulated in songbirds, hummingbirds, and parrots in the song nucleus that produces the song. And in the quail and the dove, non-vocal learning birds, you don't see that downregulation. And in the human motor cortex, these two white signals here, you do see this downregulation in two laryngomotor cortex regions that correspond in exact coordinates here on the right to the areas of the brain that have the highest activity when we humans speak. Firing patterns in the laryngomotor cortex was the work of Eddie Chang's lab, where they looked at patients undergoing surgery for removing of epileptic brain regions and found these two red spots here are showing the highest firing rates with speech production. They happen to correlate with the same areas we found downregulation of these genes. And so what's causing the downregulation? And here in the slide here on Neuro D6, shown in the songbird brain and the human brain, we looked at the regulatory regions of some of these genes and looked at their, what we call the regulatory signature. That is that part of the genome that is regulating this gene, Neuro D6, have high levels of activity or low levels of activity in the song area of songbirds. And we can see in songbird RA, it has high levels of activity, where outside of it, it has low levels of activity. And we think that's causing the differential expression of the downregulation inside the vocal learning nucleus compared to the surrounding motor circuits. And we also find that the region showing high differential activity in the regulatory region is actually a binding site for the testosterone receptor. And singing behavior in these song learning birds is hormone dependent. The more testosterone a male bird has, the more he likes to sing. And so we think this could be controlling somewhat not only the activation to sing, but maybe something about the special properties of the circuit. And it turns out, by the way, that in human brains, we do have hormone receptors in our speech areas. And why that's the case, that's interesting. Maybe it influences our courtship behaviors as we use our voices to sing arias and love songs and so forth. That's one hand-waving hypothesis I have. And so the mechanism of how that could occur is shown here on this slide, where we think in hummingbirds, parrots, and songbirds, the site that regulates the Neuro D6 protein or gene expression has acquired mutations in blue here that has allowed the androgen receptor to now bind to this region and suppress the expression of the Neuro D6 gene, such that it's downregulated in the vocal learning circuit. And when it's downregulated, Neuro D6 cannot go and bind to the regulatory region of the SLIT1 gene, and then that will cause it also to be turned off in the vocal learning circuits. And shown here is how do you turn it off in the vocal learning brain region, but not turn it off in the surrounding motor area that's controlling hand movement, for example. And what we think is going on there is in this lateral archipelagian region surrounding the RA circuit, shown in brown here, is that there are histones binding to the regulatory region of this newly evolved regulatory region, and it prevents the androgen receptor from binding to it, so you can't turn it off in the surrounding brain circuits, but you can only turn it off in the speech circuits. So, and what would that do? Shown here is that direct connection now that I talked about from the laryngomotor cortex projecting down to the brainstem circuits that control the larynx here in humans and a non-human primate, and non-human primates only having the indirect projection, but not the direct one. So in this next slide, shown in more cartoon form, this is what we think is going on with these genes. In non-vocal learning species like the dogs, like the chickens, or in the surrounding brain circuits as well for the motor behaviors, shown in green here, we have high levels of SLIT1. It's not downregulated. And when these axons from the cortex go down to the brainstem and try to synapse onto the neurons that control the voice, the vocal organs, they have high levels of the roboreceptor. And when SLIT1 binds to its receptor, it prevents the connections from forming. It stops it. However, in the vocal learning species, the SLIT1 has been turned off. And when those axons from the cortex go down to the brainstem circuits, the ROBO1 receptor, even though it's high here, it won't prevent the connections from forming. And now you'll get a direct connection to the neurons that control the muscles for voice. And so we like to actually test this hypothesis. And one way of doing this is by upregulating the SLIT1 gene in the songbird RA nucleus and see if you can make a direct projection, indirect, and break the vocal learning circuit and prevent vocal learning. Conversely, we'd like to downregulate the SLIT1 gene in a non-vocal learning bird species here, or some other species, and see if we can take a indirect projection and make it direct, and also take over a pre-existing motor learning circuit. For mammals, we like to do that in humans and non-human primates, but that's difficult, as you can imagine. But what about mice? Mice, there's a lot of work on mice for genetically manipulating genes in them, including human genes, or human versions of the genes and putting in them. But no one actually knew if mice were vocal learners or not. We assumed that they were, and no one really had studied the vocal learning circuit when we started looking on the vocal circuit in mice when we started studying them. So, in this last part of my talk, I'm going to tell you some experiments we tried on mice, taking some of these genes that we've looked at in the vocal learning birds and in humans, and leading to a hypothesis that we're calling the continuum hypothesis. When we started working on mice, a colleague of mine, Tim Hawley, found that mice have these ultrasonic songs, actually, that they produce in a very high pitch range that we can't hear. That's why it's called ultrasonic. And I'm going to play it to you, and this is what it sounds like. Sounds like a songbird. That's quite remarkable. And here's a male chasing a female, or basically trying to court her. And the more varied the sounds that he produces, the more sexier the songs sound to her, and the more she will likely court with him. And what Tim's lab found is that even different brothers from the same parents will produce different syllable types and different combinations, or let's say repertoire composition differences, like the down-up DU and the down and the high-pitched syllables here. And so my lab and several other labs study these vocal behaviors of these mice, and we've come to the conclusion that they are not vocal learners, even though they sound like a songbird song. But there is one group saying that there's no plasticity, no ability to modify the vocalizations at all in these mice. And some others, including my own lab, says they have limited ability to modify them, but they're certainly not like a human, but they're not like a parrot or a songbird, even some of the simplest songbirds. But nevertheless, there's some plasticity. What about the forebrain circuits? Well, placing neural tracers in the muscles that control the vocal behavior in mice, using transsynaptic tracers, we found that they actually have a forebrain circuit or forebrain neurons that's in the brain area where we would expect to find vocal learning circuits. And that same brain region also has activity-dependent expression of genes. When these mice sing, these genes light up, like we've seen in the songbirds. So that was a surprise, because before this finding, we did not think that mice even had a forebrain region. We thought it was all the brainstem circuit. However, when we asked if this forebrain region makes a direct projection or indirect projection to the neurons that control the vocal organs, we find that shown in these little black lines here, synapsing on these brown neurons, brown labeled neurons, is that they have a direct connection, but it's very sparse. It's 1 to 2 to 3 axons per motor neuron, as opposed to hundreds, or in humans and the song-learning birds. Further, when we looked at the specialized gene expression as we see in humans and songbirds, like downregulation of SLIT1, we can't find that downregulation here in the mouse motor cortex. This led us to the continuum hypothesis of vocal learning, where we argued that vocal learning is not a binary trait. It's not dichotomous. Rather, it's continuum. where you have simple vocal learners or animals who don't vocalize at all, like lizards. And then you have some of the simple vocal learners like mice who can change the pitch, change whether or not they produce one syllable type more than the other. And then you have the actual vocal learners themselves, like bats, songbirds, humans, dolphins, and then the complex vocal learners like humans and parrots. And I'm not saying it's a continuum like a linear continuum. It could be a stepwise continuum here. And what this would mean is in mice, we would actually not need to actually induce a vocal learning circuit with them by placing human genes into their genomes and into the brain, but enhance an already existing circuit that has some properties of what you see in human, is just not advanced enough. And this is what we're trying. And we have now actually gotten SLIT1 downregulated in the motor cortex of mice. And guess what? These mice, shown in this plot here, are producing more complex syllable types as adults than in their wild-type counterparts. We haven't gotten to the point yet we can ask, have these mice learned anything new, or can they learn something? That'll be our next experiments, but we think we're going to have to manipulate more than just one gene, like this SLIT1 here. We're probably going to have to manipulate a subset of those 55 genes. What's interesting is that these genes are interacting with each other. SLIT1 is also regulated by another gene called FOXP2. FOXP2 is famous for known mutations causing speech deficits in humans. And here's an example of a child with a FOXP2 mutation. She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
How old are you? And she says 4. So Laura is a 4-year-old child that has a mutation in this gene. She has very good auditory comprehension learning because she can hear this person asking these questions. But she has difficulty producing learned speech sounds. Yet her siblings, who are around a similar age, older or younger, are much better at producing learned speech. And so what's going on here? In a collaboration with Simon Fisher, we put the FOXP2 mutation in mice, the same one that Laura has, and we find, compared to wild type, the wild type mice, when you place females in front of them, they will produce more complex sequences or syllable types in their songs than when they just say smell a female's urine. In this bottom graph here showing more simpler songs. But when they have the FOXP2 mutation, like Laura, who has difficulty switching to words with more complex sequences, these mice have difficulty even switching to innate sequences that are more complex. So we think FOXP2, which manipulates SLIT1, was already being used to modulate vocal circuits even before speech came along. What about the brain circuit itself? These mice, they still have these layer 5 neurons shown in green here that project down to the motor neurons that control the voice with a sparse projection nonetheless. But they're more spread out in the cortex, shown in this red line here, compared to wild-type mice where it has more localization of the neurons in the motor cortex. So to finish up here, where are we at? We have now found that humans and song-learning birds have a lot of parallel connectivity and brain regions, not totally the same, but lots of parallel in their forebrain circuits here. Some of it is inherited from a common ancestor, like the auditory pathway involved in processing sounds, and some of it is novel, like the brain regions for producing learned speech. And we know a lot more about these circuits in song-learning birds, shown on the right here, even down to the individual cell types and to their firing patterns and their connectivity and the molecules that we make. So we can learn from songbirds now at the individual neurons, at the individual genes, predicting how, when a certain cell or a certain gene has a disorder to it, how that will impinge upon understanding disorders of human speech and how to repair those circuits. And going beyond humans and songbirds, shown in this phylogenetic tree of all vertebrates here, I argue that this is a common origin hypothesis, or that is, there's a deep homology in these green labeled regions, this cortex, out of which in humans and some birds, the brain regions for song and speech evolved in yellow and red here. And I argue that over the next, let's say 500 million years or less, some other species could evolve vocal learning circuits, and this is where they will be located. I think vocal learning can evolve in a limited fashion, and this is the prediction here. But it's remarkable, like the evolution of wings, how a behavior that controls speech can occur in a similar way. And so this is how we can explain, shown in this diagram, how eventually we can try to get a monkey to say the word apple like a parrot can. So the take-home messages are complex behaviors can evolve multiple times from deeply homologous but diverse brain circuits, cells, and genes. A trait like spoken language can be understood from studying distant related animals to us, like birds, more so than even non-human primates. And convergent evolution of mechanisms for a trait can be associated with similarities in disorders of that trait, like I showed you with the FOXP2 mutations. And I'll acknowledge members of my lab. I acknowledge them, as you saw in these slides presented, but others with lots of collaborators we had here from many different institutes that led to all these studies, as well as the funding support that we received over the years from NIH, like the Federal National Institute for Deafness and Communication Disorders, National Mental Health Institute, National Institute of General Medical Scientists, as well as the Neurological Disorders and Stroke, as well as NSF and Howard Hughes Medical Institute, the Packard Foundation, the Allen Institute, and the universities that I have attended. And I'll go ahead and stop there and take questions. Thank you very much for your attention.
Thank you, Dr. Jarvis, for a wonderful talk. I'll pass the mic to you right now, and you can unmute yourself and turn your camera on.
You're welcome. I'm ready to take ready to take questions. And it was interesting the first time I've ever seen myself a whole hour talking. And it was interesting to describe these slides without pointing to the actual objects. So if you didn't understand anything, please go ahead and ask. I will also say that I'm glad to be back, even if online, to the MBL. I've been up there a number of times for various things like the spines course and neuroscience course, as well as a lecture like this in 2008.
All right, thank you again. We'll get started with some questions here. First question is from Nantucket. How does the learning of sign language in gorillas fit into this question?
Yeah, so the areas of the brain that, there's a little bit of debate on this, and I'll just say which side of that debate I fall on, but there's a question about the areas of the brain that control communication with the limbs, so sign language, is either part of or overlaps with the areas that control speech or directly adjacent to it. And I argue, based upon my readings of the literature, I think it's more or less directly adjacent to it. And I think this area that controls learned movements of the limbs is basically a parallel circuit to the spoken language circuit. And the reason why chimps or non-human primates have that ability, not to the same degree as humans, but at least more advanced ability to do sign communication than they do for learned vocal communication, is because that is a pre-existing circuit out of which I think at least contributed to the evolution of the vocal learning circuit.
Great. Next question is from someone listening in from Japan. They ask, is there any connection to the cerebellum co-evolving specific for vocal learning circuits, since the role of the cerebellum in precision and learning of motor programs has been indicated?
Yes, for a long time, it was thought that the cerebellum was not involved in, no problem. For a long time, it was thought the cerebellum was not involved in anything related to speech or comprehension of speech. But in the last 10 or so years, there's been a number of studies to show that cerebellar damage, lesions, or things that affect cerebellar function, depending on the part of the cerebellum, will affect speech learning. So we do know it's involved. And many years, it's also is the question came about as to whether or not this was the case in songbirds. And nobody for a long time had found any kind of link of the cerebellum into vocal learning circuits in songbirds. But recent work has demonstrated there is some connectivity and some parts of the songbird cerebellum that also participate or contribute to vocal learning. But I don't think, though, that the cerebellum part of speech control is a uniquely evolved circuit. I think it's mostly in the forebrain itself, and an ancient part of the cerebellum has now become attached to that function as opposed to a newly specialized, evolved part of the cerebellum involved in speech.
Great. Next question. Do we have any idea if whales and or other cetaceans have these speech or song areas?
Yeah, that's been a big question for us and for many people. And actually some of the initial studies done on whales came out of students at Rockefeller University back in the 1970s. And so a good amount now is known about their vocal behavior studied over the years. But as you can imagine, trying to study a whale brain or even a dolphin brain bigger than the size of human brains is challenging, and even bringing them into the lab. Nevertheless, I have actually a graduate student who's now looking into this, where she's getting dolphin brains from strandings, and we're looking at whether or not these dolphin brains have a convergent gene regulation of the patterns that we see in humans and songbirds. And I don't know the answer yet. I wish I knew, but I can tell you that that work is ongoing, and hopefully within a year's time, we'll have some answers.
Great. This question is from someone listening in Mexico. Thanks for sharing your wonderful insights. Since purposeful observation is a critical feature of human hominid evolution and vocal learning is independent from common ancestors, do human observations of birds singing and mating have they led to the development of language in first small and then a more broad distribution in humans?
Can you repeat that again?
Let me see if I can rephrase that. Let me move on to a different question and come back to that.
Well, let me say what I think is going on, and then,
okay.
So at least give some answer here. There is a question. I saw another question related to this, whether or not what we do at speaking, as I'm doing now, and what we do as singing, as if I'm going to sing, something like this, or as these birds sing, right, is whether or not they are part of the same process or different. And what we know about our own brains, I believe, is there's some debate about this, but I go on the side of that the brain areas that control speaking are basically the same brain areas that control singing, used in different ways, or at least they're overlapping circuits. And the few studies that have been done on looking at parrots who actually imitated human speech and looking at the brain areas that actually control their ability to actually produce human speech sounds versus their own warble song are also the same brain areas. So I think singing and speaking are just different parts of a spectrum that's controlled by vocal learning circuits in the brain.
Great.
Okay.
Thank you for answering that. Next question, let's see. Are there other human families with speech disorders who have been identified that have been linked to neuro-D or androgen receptors or any of the other genes implicated in your studies?
Yes, Stephanie, who gave me that wonderful introduction, and has been one of the persons who've been studying some of these other genes like HAPNAP4 and others. I also mentioned SLIT1, which some mutations in humans cause speech-related autistic deficits or speech sound disorders. So there are other genes besides FOXP2. One thing, though, is the case is that it's remarkable that FOXP2 is one of the few genes that have the most specific effects on speech. None of these genes are totally specific to speech, but FOXP2 is one of the most specific. Some of the others impact other kinds of behaviors in more detrimental ways. So there's something that's converging on this gene in terms of its main function, but it's not the only one.
Thank you. Next question. When you get your mouse knockout studies perfected, a magic soup of knockouts, knock-ins, what will be the behavioral output? Will you create a mouse that can vocalize as creatively as songbirds?
Yeah, this has been a long-term, let's say, experimental fantasy for me that we're actually trying. And I don't expect, at least in my lifetime, even if we get to the point of getting mice to voluntarily modify their vocalizations, that we will get full-blown vocal learning as what we see in these songbirds or dolphins, or of course, even humans. But, I think we can get closer. And the one study that gives me hope is a study published in Science a few years ago by Gu et al., G-U et al., where they had a similar hypothesis on plexin gene, plexin A1, where they were able to, they found that in the human motor cortex, that gene is downregulated in the motor cortex compared to non-human primates in mice. And through similar types of experiments, they were able to downregulate that gene in the mouse motor cortex and allow what they now find as direct projections to the neurons that control the mouse forelimbs. And these mice actually were much more dexterous in actually eating spaghetti and doing things with their forelimbs than mice that didn't have this downregulation as what they see in humans. So that's not vocal behavior. It's mouse, hand movement behaviors, but getting a more advanced behavior by manipulating one gene in the manner that you see in humans gives me hope that maybe we can get these mice to do a little bit of tiny imitation with at least manipulation of one or two genes.
Next question, and we'll just take a couple more questions since we are past 9 o'clock. Have we learned about vocal learning regions in humans from strokes, particularly lacunar ones?
From strokes?
Yes, from strokes.
Yes, that's how the very first case study from Paul Broca back in the 1800s was learned, where there was, it was once thought to be very localized. It was actually a pretty broad lesion, but it is in the now famous Broca's area, where lesions there and other brain regions cause various types of speech production as well as perception deficits. And you'll hear me and Stephanie and others in our field that study non-human animals for these traits. We often call them vocal learning regions. And what do we call them in the human brains is, it's still a matter of further study. But when you look at the functional parallels with songbirds, I think we can safely, in some form or fashion, call them vocal learning circuits in the human brain as well that control speech acquisition, speech production, and learning, including those identified through strokes.
Thank you. Next question. Any ideas about what the selective pressure was in those species that evolved language?
Yes, there have been so many hypotheses as to what selected for spoken language in humans or vocal learning in the non-human vocal learning species. And my favorite that I propose is that, I'm just wondering, why is it so rare? Why isn't it not more common? And what I think is going on is that sexual selection is selecting initially for vocal learning. It's not the primary final goal, as we have more complex communication as we do now, is selection as well. But selection for complex songs that you don't habituate the opposite sex auditory system, and so you can pay attention to me, or I pay attention to you, or demonstrate how healthy you are by the diverse repertoire of sounds that you can imitate out there in the wild. There's some bird species out there like minor birds or African gray parrots that are copying all different kinds of sounds in the environment. And the more they can copy, some like Irene Pepperberg thinks that the more intelligent in human speak, they sound to the opposite sex. Well, what happens when you're producing all these varied
different kinds of sounds out there in the wild that your conspecifics won't habituate to. I also think that predators won't habituate to them as well. And so at least out there in the wild, I think the predators are selecting against vocal learning and selecting against speech. It's a semi-hand-waving hypothesis, but it's the best that I have to offer.
Very interesting. We'll make this our last question for the evening. This one comes from Tucson, Arizona. Can vocal learner birds learn from other species of songbirds? The question comes from thinking of bilingual brains as a human learning second or third languages.
Yeah, these kinds of studies go all the way back to the founders of this field, like Peter Mahler and Mark Nishi and Fernando Nottebohm, where, and people doing these kind of studies now as well, where there's a lot of actually overlap here with humans, and it depends on the particular species. But these birds go through critical periods of learning how to imitate sounds, kind of like we do for spoken language, where some bird species, like humans, will have an early period in life, like in our juvenile period, where they can learn new songs of their own species or even other species if you raise them with them early in life. And after they go through a puberty-like adult transition, it's harder for them to learn new songs, if at all, for some species like the zebrafinch. And so, so if you were to raise these young birds with another species at that age, or with humans at a young age, that's how you get parrots to best imitate human speech, or like a starling, is to make sure that you get them when they're young. Because if you get them when they're an adult, even if they're an open-ended vocal learner as an adult, it's going to be harder to imitate the sounds. What's interesting is that if you, let's take a zebrafinch, and you give it a choice and put it in a cage next to another zebrafinch male, and in another cage next to it, that's a Bengalese finch, a different species, okay? If you give it a choice, it's going to preferentially learn the songs of its own species during its critical period time frame. But if you remove its own species, it will learn the other species when there's only one model to choose from. So there's some interesting parallels there.
Dr. Jarvis, thank you so much for a wonderful lecture. I'm sure if you were here in person, we'd move on to the Meg's room for the reception, and you'd be bombarded by the 40 some odd questions that we haven't been able to answer this evening. Unfortunately, this is all the time we have for tonight. And I want to thank you again on behalf of the MBL for joining us tonight virtually, and again, for a wonderful lecture. We hope that we can welcome you back in person.
Sure, I look forward to being back in person when we overcome this pandemic. And for those in the meantime that would like to ask questions, you can go ahead and email. I may not get back to you right away, but I certainly will get back to you at some point if you have further questions.
And you will also receive these questions that were unanswered tonight.
Yes.
So yes, you can look through those. Thank you again, and thank you all for joining us from all over the world tonight.
Bye-bye.